Right, okay, I put together a video of this first part of Key Area 2.6. I put together another one for the second part, but just so I don't get my video too long, so let's give this a wee go. Right, first of all, a wee reminder of the three domains of living things. So we've got our archaea here, and they are single-celled, they've no nucleus or organelles, and while they maybe seem similar to bacteria, they've got some metabolic pathways that are similar to eukaryotes. Some have been classified as extremophiles, and we've got examples of that, like our bacteria that produce the enzymes that are used for PCR. So they exploit niches that are at the extremes, and that gives them some sort of potential to be used in industry. Next we've got our bacteria, and they're divided into groups, 3 main groups, or sometimes there's 5 main groups. You don't need to know all that kind of detail, but an example of that would be something like a rod-shaped bacilli. They're called bacilli because of their shape, or round shape, they're called cocci. And each of these groups are divided further into species level, and within that species there are different strains of bacteria. So it's just some of the terminology. Now we use bacteria to produce things like yogurts and cheeses, and they're used as well in genetic engineering. Our fungi are eukaryotic cells, so the fungi that we're kind of thinking of are things like our yeasts or molds. Some fungi are beneficial, some are harmful. Sort of beneficial fungi we could think of are things like decomposers, yeasts that are used to make bread and wine and beer, and yeasts that are used, or fungi that are used to produce antibiotics. Now we've also got, as well within the eukaryotic domain, things like protozoa and algae. So protozoa are unicellular eukaryotic cells. They've got organelles, algae are plants. They're often unicellular, and you find them growing in a wide variety of moist habitat. Well, why do we use microorganisms? Well, they're pretty adaptable, and so they can be grown, and they can produce lots of substrates from their metabolism that are useful. They can be found in all sorts of niches. I've got here deep sea vents and very cold conditions, the human guts. They're used widely in research and industry because they're easy to grow. They reproduce fast. What you can grow them on, the substrates, is often pretty cheap. Maybe a waste product from something else, and their metabolism can be controlled, and it can be changed and manipulated pretty easily. Okay, here are the sort of things that will affect microbial growth. If you've got any contamination there, then you may end up growing something that you don't want. Also, you're going to be using growth media. If it's an organism that doesn't photosynthesize, you're going to have to give it food and nutrients and vitamins that are required. Availability of oxygen is going to affect how your microbe is going to grow, and also temperature and pH. So we're going to have a look at each one of these individually. So ways of preventing contamination, well, you can use heat to do that, and the sort of thing that you would use for that would be an autoclave. So an autoclave is a device that allows steam to enter into it. It's a sealed unit, and you have that steam under pressure, and you can produce a temperature of around about 121 degrees Celsius, maybe for something like 15 minutes. And that heat is what kills the organisms. You get that higher temperature because you've got the steam under the pressure. It also means that you can kill off spores, bacteria spores, and spores are bacteria when they've gone into a dormant form, and usually what happens is those aren't killed off at lower temperatures. There's a process called pasteurization as well, produced by Mr. Louis Pasteur, and it was originally used to control the spoilage of wine, but it's now used mostly for sterilising milk, and it's involved just heating up to around about 71 degrees for 15 seconds, and then the milk's cooled rapidly, and that kills off any harmful organisms that are there, but it's not heated up so much that the proteins in the milk are affected. Radiation can be used for sterilisation, so things like UV or microwaves, and these can be used to sterilise. Microwave uses thermal effect to kill this bacteria. UV waves affect the DNA. You can use filters as well, or you can use chemical agents. So for example, chlorine is used, and it's used in particular for things like controlling Legionella, or you can use disinfectants and antiseptics. Microorganisms, like all living things, need to have energy, and if it's something like a unicellular algae, it can get its energy from photosynthesis, but other organisms need more complex compounds to be added to growth medium, and that can include things like vitamins, fatty acids, and various carbohydrates. You probably already know that microorganisms are grown in culture media, and that can either be a solid or it can be a liquid preparation, and it's got all the nutrients that a microbe needs. If it's a liquid medium, it's called a broth, and if it's solid, it's agar jelly, and you've come across agar jelly before in the laboratory. You can have a growth media which is composed of specific substances depending on the microorganism that you're growing, or you can have more complex ingredients such as beef extract. You've got to make sure your media is sterile before you use it, because obviously if it's already got other microorganisms growing in it, then that's not going to be good. You want to grow the microorganism that you're interested in. There are two types of media that you need to know about. One is called complex media, and it's got a mixture of ingredients in it where it's not the exact chemical proportion or composition and the components aren't actually known. There's a rough idea of what is there, and examples of that would be your nutrient agar that we usually use in the school laboratory. It's got maybe sometimes it has meat or yeast extracts added to it. But there's also a defined media, and they're sometimes also known as synthetic media, and they're kind of more, the components are more known and they're thought of as maybe relatively pure. So here's some examples of the types of media you might come across. So the example that I was talking about earlier, complex media, an example of that's a nutrient agar. That's the agar that you've usually got in the school lab, and most or lots of bacteria will grow on that. More specialised types of media, defined media, and there's one here called M9, and the bacteria that you would grow on that would be your E. coli or Escherichia coli. There's enriched media, and that's an example of that would be something like blood agar, and as you would expect, that means it's got blood in it, usually something like sheep blood or horse blood. And the type of bacteria that you're going to be growing on that would be bacteria, this one here, it's called Streptococcus pyogenes, and it's the sort of bacteria that is called hemolytic. It breaks down red blood cells, and it's the sort of bacteria that would be found in your throat, causes strep throat and things. You've also got this media here, it's called McConkey agar, and it's got biosalts in it and other items. And the type of bacteria that grows on that, well, here I've got something called gram-negative bacteria. Bacteria can be divided into gram-negative. Gram is a stain, a type of stain, and if it's a negative bacteria, it doesn't take up the stain. If it's called gram-positive, it does take up the stain. It's dependent on what the cell wall of the bacteria is made up of. And the sort of bacteria you get growing in that would be bacteria that grows in your intestines. Well, generally what is in your growth media with things like carbohydrate, amino acids, nucleotides, sometimes there might be some specific vitamins or fatty acids. And also as well, you need to make sure, as I mentioned before, that all media that is used under aseptic techniques. It's kept sterile to avoid contamination, and you might, in environmental situations, you might use computer monitoring to control the environmental conditions. We'll have a look at that in more detail as we go. So far we've been kind of thinking in terms of growing bacteria possibly in a laboratory, but if we're talking about getting useful products from microorganism, then we're talking about producing them or growing them on an industrial scale. And to do that, we grow them in fermenters, and this is an example of 2 large fermenters that are here. You can see there's sort of dials on them and various pipes, and there's some technology that's side here. So growing a microorganism on an industrial scale has got these large computer-controlled fermenters, and there are various sensors that make sure that the conditions there are kept at an optimal level, and that way you can produce the maximum yield of whatever product it is you're looking for. So here's a diagram then showing the idea of a computer-controlled fermenter. So factors that would affect growth would be things like your temperature, your oxygen concentration, your pH, your glucose, and various other nutrients that are needed for the microbe that you're going to be growing. And so you would have an air supply coming in that would be providing your oxygen, and you would think that would be fairly straightforward, but you would need to pipe that in probably, and in order to deliver oxygen, and you'd also have stirrers there that would help to ensure that your oxygen is mixed round and stirred for all the to make it available for all the microbes. The temperature and the pH would be monitored by probes that are connected to a computer, and that would adjust the temperature as required. So you would have a jacket that would allow cooling water in if it was too warm, or you would allow heated water in if it was getting too cold, or vice versa. And you would also be supplying nutrients and microorganisms to the microorganisms. And hopefully at the end of all of this, you would get your product, whatever that product is going to be, whether it's an enzyme or a vaccine or an antibiotic, whatever it is that you're looking to be producing. Okay, talking about some of these things in a wee bit more detail, you would think that all microbes need oxygen, but not all of them do, and there are some microbes or some microorganisms that oxygen proves fatal to them. So a microbe that must have oxygen present in order to get energy from foodstuffs is called an obligate aerobe. In other words, it needs oxygen for respiration. An organism that would die if there was oxygen present is something called an obligate anaerobe. And the thing with those organisms is they don't have the enzymes needed to break down various dangerous chemicals. So peroxides, for example, that are made in aerobic respiration. Things that, remember our catalase that breaks down our hydrogen peroxide. Some microbes, that is yeast, can grow in the presence or the absence, or especially yeast can grow in the presence or absence of oxygen, and these are called facultate anaerobes. Although they can grow in the absence of oxygen, they actually do grow better when oxygen is present, but they have the ability to grow in either conditions. Microorganisms can usually grow in a range of temperatures, although there's some sort of temperature in which the organism grows best, and that's known as the optimum temperature. Remember that temperature, we're talking here about enzymes are usually involved here, and so the optimum temperature for the enzymes is where you're aiming for. But there are some microorganisms that live in extremes of temperature, and they can survive and grow despite the temperature, and we'll have a look at the terminology for some of these, because there's some new words to be introduced. Okay, so here's the name of some of these organisms that are found at different temperatures. So some new names to be introduced. So we've got first of all here the psychrophiles. They're found in places like the oceans or the north and south poles, and they're adapted to live in very cold temperatures. Some of them have got special antifreeze proteins in the cell membranes that stops the cell membrane becoming solid. And then we've got our mesophiles, and these are normal or live at normal temperatures, room temperature, or they live in the human body. And then we've got our thermophiles or extreme thermophiles. So they're microbes that can live in very hot environments, things like volcanic vents, compost heaps, and hot springs. So lastly, when you're growing your microorganisms, you need to think about pH. If you're growing fungi, some of them tend to prefer a more acidic environment, bacteria, more neutral environment. So we tend to use things like vinegar or citric acid to preserve food because they stop microbes from growing. So if we're growing a microorganism for some sort of useful product, you have to keep the pH relatively constant, and buffers are used to do that. And now that's the end of this first video on this key area 2.6. You can go to your textbook, and if you look at page number 189 and read to page, I'm turning over the pages as I'm doing this, 195, and there's some questions there as well on that page there, but you can have a read of that and answer the questions. Thanks very much for listening.